Hello, this is Doug Sparks from M2M Technologies, and I'm going to be talking about biosensor commercialization strategies and kind of give some guidance to startups, particularly in the biomedical device area. Now, a biosensor, it's more than just technology. So when you have a startup company, a lot of times they're engineers, they have some cool technology, and they think they can commercialize it. But it really takes a lot more than that. I mean, it takes money, fundraising. That's the CEO's constant job. You have to have great quality, have to have intellectual property. You probably have to have software, packaging your device. You've got to think about FDA approval if you're in the US. If you're trying to get insurance coverage for your customers, you're going to have to think about reimbursement strategies. How do you manufacture it? Where do you do your research and development? Talking to venture capital firms, doing an IPO, how do you exit and sell the company? So those are all the kinds of things that you, in the back of your head, you got to be conscious of and kind of be working on these things in parallel throughout the commercialization process. Now, there are a number of ways to track to make sure you're not missing anything. I've been using kind of a 4-stage product development process that I used a long time ago, first in the automotive industry, then in medical devices as well, and industrial devices. And if you think of it, you can think of it in terms of the TRL numbers. I put those down on the bottom, but generally I have a concept stage where you've got this device idea. You might be forming some IP. If there's a medical device, you got to be thinking of regulatory and legal issues. Got to start filing your patents, and then thinking about material issues, particularly biocompatible materials. You'll then go into the development stage where you'll actually make kind of a working part, a functioning part. But in addition to doing that technical aspect, you want to put together a business plan. You might want to do risk analysis on the medical device. You might want to start talking to people about the regulatory issues. What classification of device is it going to be? Is there a reimbursement path? You might start doing some of the basic material and biocompatibility tests. Maybe think of if it's going to be implanted or in the bloodstream like a drug infusion device. How are you going to sterilize this, and will the sterilization technique destroy the device? Okay, then you go to the prototype stage. That's where you make a device pretty much like what you're going to sell. And some of these steps are generic to everything you would develop, but in red here I've highlighted some of the things in the prototype stage. Is going and talking to an FDA regulator during the prototype stage to make sure your device satisfies what they're thinking. And particularly if there's a predicate device, this is a good time to do that. Search for your predicate device, rationalize why this predicate device is very similar to your invention or your new product. If you require animal testing, this is the time where you start thinking about that. You may want to conduct some animal tests, pilot animal tests on an implantable. You'll want to get fully involved with the quality system, ISO 13485, and start doing audit planning. And then finally, as you go into production, you've got to ramp things up. So you're going to have to be very particular on the quality of the products you're making. You're going to have to complete your animal and clinical trials, and then eventually work with manufacturers. This is probably when you're going to start doing strategic partnerships, particularly for manufacturing and distribution in the medical industry. So this is just a few items in the overall product development process that you need to talk to someone about, and that's what M2M Technologies can help you out with. Now, at the end of each one of these sections, concept development, prototype, manufacturing, you need to have an exit checklist. And that's where you get your whole team together, and that's legal, business, operations, engineering, regulatory, And you got to go through a checklist and make sure everybody agrees, yeah, we've completed everything we need to get out of the development phase, or we completed everything we need to get out of the concept stage, and it needs to be signed off by everybody. Early on, you can have one or two things that are still a no, and you can address those in the next phase. But the farther along the process, you've got to make sure everything, particularly when you get out a prototype, everything has to be a yes in this checklist. Another form that you often see in sensor development projects and biomedical sensor projects is a PFMEA or a DFMEA. The DFMEA is the design, failure mode, effects, and analysis, I guess, approach to looking at medical devices, to think of everything that you're designing into this product, how they might interact, how they could fail, and then come up with ways to prevent failure. That's the whole thrust of this. The same can be said for a wafer process. If this is a micro machine wafer, you go through the whole wafer process and do the same thing of process, failure mode, effects, and analysis. What part of the unique wafer processing steps can cause early failure in the device or low yields? Now, ISO 13485, that's the medical focused part of the ISO 9001-2015 system. It's very similar. If you can pass your 9001-215 ISO audit, chances are, with a little bit of tweaking and work, you can pass the medical device quality management audits as well. So that's what your goal is from a quality department, to be able to pass through all the stages of these audits and prove that you're making a high quality medical device to international standards. FDA approval. If you're in the US, you've got to get FDA approval. This is for medical devices as well as pharmaceuticals and biopharmaceuticals. So with devices, you have class 1, think of a Band-Aid. There's minimal risk to those in the public. Class 2, drug pumps. That's something that I've worked on, class 2 devices. Typically, you can get FDA approval with the predicate device approach on those. Class 3, high risk, implantable devices. Think of neural probes, think of pacemakers, internal drug pumps, insulin pumps, etc. Those are class 3s. Those are very difficult to get through, and you have to have the most stringent of controls and testing and clinical trials to get through that class 3 device approval. Here's some examples of class 2 devices. Typically use a 510K predicate device approach. There's a, this is a MEMS drug infusion pump flow sensor that we got approved. There's external muscle stimulators, blood pressure measurement, another user of MEMS products. Those are examples of class 2 devices. Class 3, medical implants, 3D printed stents, micro-machined Pyrex pressure sensors for cardio placement, neural stimulators, orthopedic implants with or without stensors or stents, or etc. So those are the kind of class 3 implantable devices that can cause infection, that can result in, thrombosis, etc. All kinds of biological interactions that really have to be well thought out, well tested before they're introduced into the public arena. Now, reliability accelerated testing. This is done in the aerospace industry, in the automotive industry, thinking about operating temperatures, etc. Now, in a human implant, the operating temperature is very narrow. That's good. The bad things are that, you're essentially in the human body, it's like saline solution, warm saline solution with white blood cells. It's very corrosive. You can coat things. So there's a lot of things to think of. You have to think of also electrostatic discharge, electromagnetic interference in a hospital setting, for example. And so there's a lot of testing that you've got to go through with both just saline solution soaking, warm saline solution soaking, or animal cell growth trials. Sterilization. You wouldn't think of this as a barrier, but obviously to sterilize something, you're able to kill bacteria and viruses. Some of those techniques can damage electronics or MEMS devices. Gamma rays, very common. It'll erase mini EPROMs. It'll actually turn Pyrex yellow. Autoclave, that's just, hot high pressure steam, that'll corrode aluminum and copper metallization on ICs, circuit boards. And then ETO, that's kind of an aggressive organic attack to kill the polymers, but it'll destroy a lot of polymers and biomaterials. So each sterilization method has drawbacks and has things to consider when you're doing product design and product packaging. So just in conclusion, when launching a biomedical product, you got to think of a lot of commercialization issues. You've got to think a lot of quality and regulatory issues, a lot of paperwork, whether it's FDA, whether it's DFMEA or ISO approval or insurance reimbursement. So you've got to have a systematic, well-documented approach to everything you've done to show auditors, and then you've got to think outside the typical MEMS sensor designer. I'll think outside of that box to think of sterilization, to think about cell growth, to think about corrosive environments in a biological application. So if you have any questions, please contact me. Go to our website or contact me directly through email. Thank you.